We're done. It's okay. Okay, let's go. Let's start. Matthew,
Yes.
on behalf of REBRASNI, I would like to thank you a lot for being with us tonight. Just to give a brief introduction, Matthew Sampsom is a member of REBRASNI. He's an international member of REBRASNI. He's an associate professor in Boston, Harvard's medical school, has a lot of publications on nephrotic scene and genomics, and I think he's very, very Hebrasni to say like that. It's like our, our spirit of investigating this disease and etc. And I will give for everybody here an extract of his lab. It's in the chat here. The goals of his lab. I took from your lab site, and I think that the goals are perfectly written, and I think it's a pleasure to have you with us. Thank you a lot. And Onushik would like say a word as well for you and for everybody. Please, Onushik, come on.
So, hi, Matt. I
Hi.
want to welcome you. It's great to have you among us one more time today, giving us a broad view on genetic basis of nephrotic syndrome. And in addition to what Paulo told you guys, everyone, I want to say that Matt is not only a REBRASNI member, but he is with us in REBRASNI since its very beginning. And he has significantly contributed to our main plans so far, and to both of the publications that came from our group so far. So he's really into the full story of REBRASNI and within the full spectrum of activities of our group. So welcome one more time. It's great to have you here.
Thank you so much. And really thank you for the warm welcome. And I'm glad that we all feel the same way about each other. I feel like a full member of not only the research network, but the family. Um, Before this world of COVID, I had a wonderful series of visits with you all. Got to see many places, many institutions, meet many friends and colleagues. And I'm just very much looking forward to the day when the travel restrictions are lifted and we can really launch and accelerate the research that we've started a number of years ago. So again, thank you for the invitation. And I just want to, I'm not the only person from my group joining today. I have one other member of the group joining. Dr. Anna Onushik is also here from my group. So you'll have a couple people from the Sampsom lab here. Okay, so let me try to share my slides here. And oh, someone raised their hand. Anna, you raised your hand. Now you put it down. Okay. Okay, so let's try to share my screen here. And let's see. You let me know if I can find it. Let's see. Give me 1 second. Oh, okay. Open recent. See, I'm like this expert on nephrotic syndrome genomics, but I don't know how to use PowerPoint. You know, I'm like a medical student. Okay. Okay. I'm gonna share the screen here.
Very good.
Okay. Okay, ignore. Ignore. And okay, how's that look? Does it look okay?
Yeah, it does.
Okay. So I can't see anyone raising their hand or anything for some reason. So just if you have any questions, just let me know, okay? All right. So I want to talk to you guys about genetic architecture of nephrotic syndrome. And the objective of this is I want to talk about the genetic causes and contributors to nephrotic syndrome within this framework of this concept of genetic architecture. Because if we understand things from the perspective of genetic architecture, it lets us be very fluid in our conversations and our conceptualization of this. So what's genetic architecture? Well, I define it, and others define it, as this overall composition of variants influencing a given trait, here nephrotic syndrome. And we're talking about variants that are some of the parameters are the number of variants that influence nephrotic syndrome, the effect size from small effects to large effects, the allele frequency of these variants in a population, the way variants interact with each other, and the way that variants interact with the environment. So again, if we think about nephrotic syndrome as in terms of genetic architecture, we don't have to focus only on Mendelian or APOL1. We can think about all of the genetic contributors. And it's really my belief that every single patient that we see who has nephrotic syndrome has a genetic component. Siri's talking. Every patient with nephrotic syndrome has a genetic contributor, but the genetic architecture differs among patients. So I wanted to also just highlight efforts for genomic discovery in pediatric steroid-sensitive nephrotic syndrome, because it's a form that we don't often consider in terms of genetics, but I think it's actually quite important, and worldwide efforts are helping us to understand. So a lot of the work here that I began when I was at Children's Hospital in the late 2000s was driven by my frustration with our classification of nephrotic syndrome. We classify patients with these imprecise descriptions, such as histology. So if a patient has minimal changes on their light microscopy, we say they have minimal change disease. Or if they have focal areas of glomerulosclerosis, we say their disease is FSGS. If we see IgM or C1q on immunofluorescence, that's the name of their disease.
Excuse me, Matt.
Yes.
I don't know if it's happening for everybody, but I can't see the slides changing.
It's not changing.
I think you have to
slide.
full presentation, something like that.
Okay, hold on. The slides are not changing. Okay, let's see.
Yeah. I think you should put on full presentation, something like that. I don't know the name in English, how to show
present now. Present a tab. Present, no, cancel. Present, I thought I did a window. Let's try this again. Let's walk through this. Make sure that I'm doing it okay.
Okay.
So I'm going to share this. And how's that look right now?
It looks the same because it's not full presentation yet.
What if I swap displays like that?
It's not changing.
I guess you have to go to full screen.
Yeah, I do that on my PowerPoint. It's totally fine. Why is it doing that? Let's see.
Because here it's showing your screen like the
I see it.
in the PowerPoint.
Okay. Let's see. Now you see it like that?
Yes.
Okay, that's kind of lame, but let's see what happens if I do it like.
What if you press F5?
F5. All right. F5. No. Let's see if I do it like. What happens if I do it like that? Do you see anything changing?
No.
No, it's the same.
Interesting. Okay. Can we do it from this perspective? Is this good or is this not big enough?
I think you can try, and if it's not big enough, we can tell you.
Like, we're all family here, right? You just let me know. We'll just sort it out, right?
Yeah.
All right. Just we'll do it together. We'll be a team like we always are. Okay?
Okay.
All right. So we're going to talk about, so you didn't even see this, right?
No.
Oh, yeah. Oh, my gosh. So embarrassing. So
it happens for everybody in every.
I know, but I'm like a Harvard professor or something. I'm supposed to be very impressive, What did my boss say? Okay. So we'll talk about genetic architecture, and we'll talk about steroid-sensitive nephrotic syndrome. And we all recognize, all of us know that if we say a patient has MCD or FSGS, that doesn't really tell us at all what their molecular signature is. And how can we give a medicine that treats MCD? What we really want is a medicine that turns off a pathway that's too high or turns up a pathway that's too low. And when we talk about response to immunosuppressants, that's even more confusing, right? You know, we say, oh, our patient is steroid sensitive, but they're an infrequent relapser. Oh, wait, no, they're a frequent relapser. Now they're steroid dependent. Oh, now they have primary steroid resistance or secondary steroid resistance, but sometimes they're a late non-responder. This is not this is somewhat helpful in terms of clinical trials that give different therapies based on these classifications, but it's not ideal. We want molecular diagnoses, and we think that doing it through genetics is the best way to do that. And again, the point I want to bring up for you guys down in Brazil actually from a genetic architecture perspective is very, very similar to the United States, because neither of our countries are, in our countries, people who have unions and have babies together are typically not related to each other. And they come from African backgrounds and native South American backgrounds and European backgrounds. So this is not a highly recessive set of diseases. We have patients in populations, and the familial disease is less than like in the Middle East. Of course, as nephrologists, we want to take the best genomically informed care of every patient that comes to see us. So in some situations, we need to do family-based discovery or diagnosis to find patients who have Mendelian forms of disease. But until recently, we didn't have the ability to understand the genetic signatures of those patients who don't have Mendelian disease. So work that was led by your group that was published a number of years ago when I first got to meet you all, you guys showed that about like 8 to 10% of Brazilian patients with end-stage kidney disease had a Mendelian form of disease. Okay, so what about the other 90%? What I want to tell you about is the genetic architecture is such that of those 90% of patients without familial disease or Mendelian disease, we are starting to understand genetic signatures. Does that make sense? Cool. So the goal of our of my research group is to discover known and novel nephrotic syndrome associated genetic variants. And if we get these genetic variants in our hands, we can do two things. More immediately, as a clinical epidemiologist, we can figure out the clinical associations for those who have genetic signatures of disease. What are the associations in terms of age of onset, eGFR at baseline, progression of CKD decline over time? And this is, you guys have done amazing work that's been published about this in terms of APOL1 and Mendelian disease, really these clinical epidemiologic associations in Brazil. And we do that too in the US and abroad, right? But we're also interested, I'm greedy, okay? I don't want to just do associations. I want to find treatments and cures. And to do that, we have to understand mechanisms, just like you guys want to do. So if we find a genetic variant, we can try to do the genetic mapping, and then that leads to mechanisms and ultimately medicines, all right? So we have to define the biology experimentally and identify targets for therapeutic intervention. So that's what that's all we're trying to do, figure out the genetic basis to lead to epidemiologic associations and mechanism. So again, when we talk about the genetic architecture of disease, of nephrotic syndrome or any disease, if you're gonna not remember any other slide, just remember this one, that every allele that's associated with nephrotic syndrome, we can characterize it on two axes. That disease allele of the population, ranging from very rare or absent to common in the population, like in 5%. And we can think about the effect size, like an odds ratio, maybe 1.1 increased odds of disease versus a 50 times increased odds of disease. And you can see we can map our knowledge of nephrotic syndrome or genetics onto these axes. So Mendelian forms of nephrotic syndrome arise from alleles that are very rare or novel in the population. And when they're present, they're essentially fully penetrant and have a very, very high effect size. By contrast, GWAS variants, things that we've discovered for steroid-sensitive nephrotic syndrome, these variants are common in the population, and they result in a lower effect size, which means not every single person with this genetic variant gets the disease. There needs to be some sort of environmental interaction or another genetic interaction. But we're very lucky and fortunate in nephrotic syndrome research to have a group of common genetic variants that have large effect sizes. And if you were my medical students, I would at this point pause and make someone give me the answer, but you're not my medical students, you're my colleagues and family. So I'll just say APOL1 and genetic variants associated with membranous nephropathy. These are common in the population, especially in African ancestry, APOL1, but they have very big effect size. And the same thing for membranous nephropathy. So when we are thinking about the genetic architecture of nephrotic syndrome that the rare Mendelian forms, those are the genetic forms that come from our study of families for the most part. Whereas the more common variants come from the study of populations. Again, showing the value that every single patient with nephrotic syndrome, if we all had unlimited time and unlimited money, we would recruit every single patient who has nephrotic syndrome across your country and mine, and we'd discover genetic contributions in each of them. And people used to talk about mutations or polymorphism or risk alleles, but in my opinion, what I suggest is that we just call them variants. Everything here is a variant. This one is a rare variant that's pathogenic. This is a common variant. This is a low frequency variant. And these variants can be risk variants or they can be causal variants. And rather than, it's very kind of loose to say mutation or polymorphism. Okay, so that's how I try to train folks in my group and here as well. So let's start. Now I'm going to kind of walk through. I'm going to use this table throughout the talk, and I'm going to go through each example as we talk about the genetic architecture of nephrotic syndrome that you and I may see in our practice. Before I move into this section, does anyone have any questions? Okay, so let's start with talking about the forms of nephrotic syndrome that were first discovered to have a genetic basis, rare Mendelian causes of steroid-resistant nephrotic syndrome, or FSGS. So this is a figure that was put together by my boss, Dr. Friedhelm Hildebrandt, back in 2016. And what this is showing is that from the late 1990s with Podicin NPHS2 and WT1 and then NPHS1, there's been an explosion of single genes here that are found to harbor rare pathogenic variants or mutations that are sufficient in and of themselves to cause disease. And the form of nephrotic syndrome they cause is distinct. It's really steroid-resistant nephrotic syndrome. And among most of the patients who get a biopsy, most of them end up having FSGS. But it's not 100% correlation, as you know. And over time, enough of these genes have been discovered that they can be clustered into different biological networks. We initially discovered, and when I say we, it wasn't me, I was like in high school or college, nephrin and podicin, and we're part of the podocyte slit diaphragm and CD2AP, TRPC6 to the calcium channel. And over time, it was actin dynamics, coenzyme Q biosynthesis, more recently nucleoporins, lysosomes. So the unraveling and the identification of the genes that harbor Mendelian mutations are informing us about the biology that's driving this condition. And for instance, maybe that will lead towards treatment. So CoQ10 biosynthesis suggests that perhaps CoQ supplementation may help ameliorate the disease. Or TRPC6 is a calcium channel, and TRPC6 blockers are now being tested in clinical trials. So knowing these gene products matter a lot. Now, these certainly matter, and they're certainly important for those individuals who have Mendelian forms of nephrotic syndrome, but we have to know what the prevalence is in the countries in which we're taking care of patients. So if a child shows up with steroid-resistant nephrotic syndrome in Saudi Arabia or Egypt or Turkey, the chances that they have a Mendelian form of disease is about 45%. But if they show up in Ann Arbor or Los Angeles, that number is probably about 15%. Or if they show up in Sao Paulo or Curitiba or Atibaia or Rio de Janeiro, it's about 8%. How did I do the pronunciation? Did I do okay? I was trying to impress you guys about all the places I've been. So
very good, very good.
Thank you, thank you. So about, obrigado, 8%. So, and it's only patients with steroid-resistant nephrotic syndrome. Patients who have steroid-sensitive nephrotic syndrome, it's essentially, Mendelian forms of disease are essentially non-existent. And I think this is an important slide for us as clinicians because just like any other lab test, we want to know what the chances are of getting a positive test, whether we're checking an ANA or a C3 or a basic metabolic panel. So if you and your colleagues know that only about 10% of Brazilian kids with SRNS are going to have a Mendelian disease, when you get 6 negatives in a row, you shouldn't be discouraged because that makes sense. I think if you think that 30% of your patients are going to have a Mendelian form of disease, maybe getting 6 negatives in a row will result in you not getting the test anymore, and that'd be the wrong message to take. And you can break this down even farther and talk about, do I have enough? Is that 8% or that's 11% in Boston? We know that that number is higher if you're an infant or in your first 2 years of life, and it's lower as you get older. So we need to be precision clinicians when we see a 2-year-old with steroid-resistant nephrotic syndrome versus an 11-year-old. We need to know the difference in prevalence estimates. So that's the form of genetic architecture of Mendelian disease. Let's talk about, we went rare with big effect size. Let's talk about common with big effect size. And that's found in membranous nephropathy and APOL1 associated FSGS. So this is a GWAS of membranous nephropathy, and a GWAS looks across the genome from chromosome 1 to chromosome 23, and it takes each dot is a variant. And let's see, hold on, let's see. Let me just make it a little bit bigger for us. Okay, so we can look here, and what we can see is if these dots, these are SNPs, if they make it above this red line, that's a statistically significant value. So in these 3 studies of membranous nephropathy, there are 2 regions of the genome at chromosome 6 and chromosome 2, which are found to be associated strongly with membranous nephropathy. And chromosome 6 is it's mapping to an HLA-DQ. Does anyone know what the site chromosome 2 is, what protein that maps to? No? It's PLA2R1, okay? So PLA2R1 is at chromosome 2, and biochemical research has found that PLA2R1 receptors, the PLA2R1 gene, results in an antibody being formed against it that leads to membranous nephropathy. So because membranous nephropathy it's quasi-Mendelian, it's caused by variation in only 2 or 3 genes, they were able to discover that in a very small number of patients. So that gives us hope that if we collect more patients for genomic research that aren't related, they don't have to be familial, that we can discover, again, genetic signatures. And especially the adult nephrologists here, you know that PLA2R1 testing, whether it's serum testing, antibody testing, has become an increasingly standard practice. So this is really translational genomics. This is just an updated GWAS of membranous nephropathy. This is now about 10 times as many cases. And you're seeing HLA and PLA2R1, but now we're finding NF-kappa B and an interferon response element over here, and we're learning these markers can be used for clinical stratification, but also for mechanistic understanding. And again, this study was led by Christoph Kierluke out of Columbia University, working with investigators around the world. And whether people were contributing thousands of patients or hundreds of patients, each patient matters, and it's important. And as you guys know, there's massive disparities in the participation of patients around the world in genomic research, and in particular, the global South is very, very underrepresented in genomic research. So the work that you guys are doing in Brazil to bring more of these patients in for genomic discovery matters a lot and is making a big impact. Then the story of APOL1 comes from the recognition that Black people in America with FSGS Black people in America have a much higher rate of FSGS than non-Black people. So there's a hypothesis that there is something in the genome of Black individuals, and we know because of admixture that Black individuals have a mix of European chromosomes and African chromosomes. The hypothesis is that the genetic factor contributing to FSGS was in an African part of the chromosome, a Black person's chromosome. So they use this, investigators use this strategy called mapping by admixture linkage, and they were able to pinpoint a region in the genome of Black individuals that harbored the risk for the disease by taking advantage of that disparity. And what they found ultimately is that it was APOL1 was the genetic risk factor here. And APOL1 is very common in the population because these APOL1 exonic variants protect against sleeping sickness. And when these variants came to be in sub-Saharan Africa 9,000 years ago, sleeping sickness was killing people before they could procreate or have more babies. So people who had this APOL1 variant were protected from dying from sleeping sickness, and then they were able to make more babies. And those babies were able to make more babies compared to those without the variant. So that's called positive selection or selective sweep. So this was a very, very beneficial variant to stay alive long enough to reproduce, but it came at a cost of greatly increased odds of developing FSGS. And what that means is that, that these studies in the US found that among all Black patients with FSGS that we see in our clinics, whether adult or children, 70% of them have their FSGS with a high-risk APOL1 genotype, which means that if we can do translational research and discover the mechanisms and treatments for APOL1 disease, then we have the opportunity to help a huge number of patients with the disease. And this is different. This is in contrast to Mendelian forms of disease, where collectively it might explain 10 to 15% of our cases, but that 10 to 15 could be made up of 30 different individual genes. So maybe one drug will help all these patients. We will probably have to make a drug against multiple pathways for Mendelian disease. And I think a really cool aspect of the research that you all have put together and published was that these prevalence estimates are very, very similar in Brazil, which implies to me that there's probably, our environments are somewhat different between, Brazil and North America in terms of profile of infectious diseases and viruses and environmental aspects, but the prevalence and the age of onset are quite similar, which to me implies that there's some commonalities here in the genetic makeup. And I think and also a key contribution that your network has made is showing that it's not appropriate to only do APOL1 testing in people who report themselves as being Black. You guys have shown that there's a lot of African chromosomes in people who don't say that they're Black or white or etc. That you really APOL1 testing should be done in all individuals with nephrotic syndrome in Brazil. And I think that's been a key contribution. I think another key contribution that you all have made is showing everywhere else in the world thus far has shown that the disease only comes with 2 risk alleles. There are many, many more people with 1 risk allele. And along with a study of HIV-associated nephropathy in South Africa, your study recently that came out was one of the only other one that I know of that shows that having 1 risk allele may have increased risks as well. And if larger studies that you guys do bear that out and it prompts people in the rest of the world to follow your example, it may be that we as clinicians start to pay more attention to patients with 1 risk allele too, as well. This is an example of what we can do if we divide patients with a high-risk APOL1 genotype and the low-risk APOL1 genotype. We did a study that asked if other genes are differentially expressed between the 2 groups, because it may not be APOL1 alone that's causing the disease. It may be APOL1 and other genes that it contributes to pathogenesis. So we did a study here that looked that each green dot here is a gene. These are genes whose expression is higher in the glomeruli of high-risk individuals, and these are genes whose expression is lower in the glomeruli of high-risk individuals. And this is the statistical significance line here, this horizontal line. And this is showing that these 10 genes are significantly different in the glomeruli of high-risk versus low-risk individuals. And the number of these genes are involved in mitochondrial function, and the number of these genes are involved in calcium regulation. So this is, oh, maybe you haven't been seeing my arrow, have you? So this STC1, I'll talk about a little bit more. So STC1 stannocalcium plays a role in mitochondrial and calcium-related processes. And working with Astrid Wines at the Brigham and Women's Hospital, she looked at STC1 staining in normal kidneys, in kidneys with collapsing FSGS but no risk alleles, and then collapsing FSGS with risk alleles, and showed that there's increased STC1 staining in high-risk FSGS as opposed to low-risk FSGS. So this is a very new finding. It's recent, and it suggests that if we do some more research on STC1, maybe we're going to find a therapeutic target, because it's unlikely, as we know, for all the other, whether it's hypertension, there's not one drug for all patients, or proteinuria, whether it's ACEs or ARBs or etc. It's likely that APOL1 associated disease will probably have to attack it with different therapeutic targets, and perhaps STC1 is going to be one of those targets. And if you want to, we really believe in data sharing. So if you want to learn more about what we did here, or you want to browse the data yourself, you can go to this portal, apol1portal.org, to search the results and explore. And of course, I'd always be happy to answer any questions that you might have. I want to briefly talk about another form of genetic architecture, which is these low-frequency variants with intermediate effects. So this was a study that compared 178 European patients with FSGS with 378 controls. And they were all European patients and all European controls. And what they did is they looked for genes in which there were significantly more pathogenic variants in cases versus controls. And using this strategy in a very small number of patients, not families, but not tens of thousands of cases, they were able to find that these genes, including COL4A4 and APOL1, and some that had never been discovered before, like CAT2B and DLG5, these harbored an excess burden of pathogenic variants. So this was burden testing. So we always have assumed that there are variants that are low frequency that increase risk of disease, but we haven't historically been able to discover them. But again, using modern methodologies and very well phenotyped patients that come from clinical nephrologists who say for sure that these are FSGS or not, we're able to find these. What's interesting is this was one of the first indications that COL4A4, a gene historically associated with Alport syndrome, was also associated with FSGS, and we've now subsequently come to realize that COL4A4 often looks like SRNS FSGS without Alport. What's interesting also to me is APOL1 became one of these top genes, and what we know is very carefully through this genetic PCA testing that these patients were all very, very tightly within a continental European genetic ancestry. But despite that, we clearly are not able to see closely enough to recognize that small parts of their genome clearly are of African descent. So again, it suggests that even in the United States, it's risky among FSGS patients to not test for APOL1 in white patients. So then I want to just finish up by talking a little bit about steroid-sensitive nephrotic syndrome. And this is an example of a disease. It's the most common form of nephrotic syndrome that we see in children. And oftentimes, it's steroid-sensitive, so it hasn't gotten as much attention because they do respond to therapy. And then the biopsies look like minimal change. And the name minimal change makes it seem like it's not a big deal. But we all know, all of us practicing clinicians, that sometimes being steroid sensitive or immunosuppressive sensitive is the worst thing for a patient because it works. So we give them steroids year after year after year, or Cellcept year after year, or Cytoxan, and they jump from therapy to therapy until it doesn't work, and then we switch, and they pay the cost with hypertension, cataracts, diabetes, bone disease, growth, mood failures. So I think it's just as important to figure out the genetic basis of steroid-sensitive nephrotic syndrome, because I would love that one day we don't give steroids, we give the appropriately genomically inspired therapy. So these are genome-wide association studies that have been done for pediatric steroid-sensitive nephrotic syndrome over the last decade, and 5 different studies. You don't have to look too hard. If you just blur your eyes, you keep seeing there's the same peak in each of these studies. Here, 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 and here. So what we're showing is, and this is in Japan and Korea, and this is in the US and Sub-Saharan Africa and Europe and European patients. This is East Asian, this is European, and this is Sri Lankan. Around the world, there is genetic changes in the HLA region that are consistently increasing the risk and contributing to SSNS in children. And this is just words that summarize this. What I want you to recognize is that very small number of cases contributed to this disease, to this discovery, excuse me. So only 214 patients were found by Rashid Gabadagis and another pediatric nephrologist to make this initial discovery. And then research in low numbers of hundreds of patients found more. What you see here, these odds ratios are pretty big for genome-wide association studies. So it means that if we could figure out what specific HLA residue or peptide binding pocket is altered, we have a chance to help a lot of patients. And what I'm also showing here is not only is it variation in MHC, but we're starting to find additional hits outside of the HLA region, including this gene called CAL-HM6, which is known to be involved in innate immunity. A tumor necrosis factor, soluble factor, again, involved in innate immunity. And one that we found really interesting that I'm excited about is nephrin. So most people here know that nephrin mutations, rare pathogenic variants, cause congenital nephrotic syndrome or steroid-resistant nephrotic syndrome, but no one has ever implicated common variants in nephrin associated with steroid-sensitive nephrotic syndrome. There's a couple small studies that hinted at it a couple years ago, but that not a lot of people paid a lot of attention to. But led by Dr. Kazumoto Iijima, another pediatric nephrologist who's a key member of our society and in IPNA, and Professor Katsushi Tokunaga in Japan, along with Pierre Ronco and Francesco Emma, and Marina Vivarelli, some more of our colleagues in the EU, and Rashid Gabadagis and myself and others in the US. We did this study that found, in fact, that this nephrin variants in nephrin are associated with congenital nephrotic syndrome. And really, the immunogenetics, especially at HLA, is very, very challenging. Work in my lab we're driving forward is really working on honing in on the specific HLA variants. But then to try to understand the immune system function and how it impacts the kidney cells. I never wanted to be an immunologist. I really am interested in the podocyte got me at first, and the actin cytoskeleton. But this is where the disease signal is at. So I'm going to try to spend the next 20, 30 years to try to figure this out with our team. But an easier one to start with is nephrin, because it doesn't involve the immune system. We know so much about this molecule already. But how do these common variants impact disease? So we looked a little bit closer, and we found that in a GWAS, the hits in nephrin are not the GWAS hits are not coding for the most part. Okay, so you can't directly implicate a gene or protein product or a loss of function, so you have to do more work. And what you can see here is these hits, these are all individual SNPs, and they fall within these coding and non-coding regions, and they fall in this very gene-dense region. And we actually don't know what exact gene is the effect gene here that's causing the increased risk of SSNS, but we have some hints. One of the hints is that all the top hits are in these 2 genes, nephrin and KIRL2. We also know that KIRL2 does have a role in glomerular health. But we also see that of these nephrin hits, 3 of them are synonymous changes within an exon of nephrin. So we wonder whether the protein of itself is being altered with mRNA. I'm gonna just briefly say that this paradigm of rare nephrin hits causing SRNS, but common ones causing SSNS, we're seeing examples like this in other paradigms in kidney disease. For instance, COL4A3 is a known Alport syndrome gene for rare pathogenic variants, but recent research into diabetic kidney disease, DKD, shows that more common variants in COL4A3 are associated with glomerular basement membrane thickness at DKD. So what I think is going to happen in the future is more and more we're going to find that there are proteins that matter in kidney homeostasis and kidney physiology. And if you break them really badly, you get rare early onset bad disease like Alport syndrome or congenital nephrotic syndrome. But if you alter their regulation or you give them, less penetrant or less harmful variants, you're going to get contributions to more complex diseases like SSNS or DKD. So we use the Neptune study, which I've been a part of for more than a decade, to try to better understand what these nephrin variants may be doing that contribute to pediatric SSNS. Well, we found that these genetic variants were not necessarily regulatory by changing gene expression. So these are all the Neptune patients who did not have the nephrin risk allele, and these are the 4 patients who had the nephrin risk allele, and their expression of nephrin was equal. Their expression of genes, other genes in that region were equal as well. So this risk haplotype did not change the expression level of nephrin. We also did a study for allele specific, for splice variants, and we found that this nephrin risk allele did not result in exon skipping. If this is the normal 3 exons, this allele did not result in an abnormal protein by exon skipping or shortening of an exon. There's no difference. But what we did find is that there was evidence for allele-specific expression. So what's allele-specific expression? Allele-specific expression says that in a normal situation, if this is the nephrin gene, and this is the nephrin chromosome from mom, and this is the nephrin chromosome from dad, ordinarily, we'd expect 50% of nephrin to come from mom and 50% of the transcripts to come from dad. But allele-specific expression is when there's an imbalance, and when you're having a preferential transcription and translation of an allele from one parent. So we did a study and we asked whether there's allele-specific expression associated with this nephrin risk haplotype, and the answer is yes. So in patients without the risk haplotype, this is a table on the y-axis. If you're down here, these patients, each dot is a patient. These patients had very little allele-specific expression. And whereas the patients with the risk haplotype, they had statistically significant increased allele-specific expression. So what does that look like? For these 4 individuals in Neptune for whom we have glomerular expression data, we have each of these is a patient 1, 2, 3, and 4. These patients had significantly less nephrin transcribed from the risk haplotype than from the normal haplotype. Each of these patients were heterozygous. So the risk haplotype came from one parent and the wild type from the other. And each of them had decreased expression of the risk haplotype. Now, why could this be contributing to disease? We need to figure that out. One reason may be that this risk haplotype is having decreased expression because it is pathogenic and toxic to the cells. So the cell has to ramp up its expression of the normal chromosome, and there's increased cellular stress. And then there's some other explanations that have to do with translational pausing and transcriptional dynamics. But if we have this statistically significant difference, what it tells us is now it's incumbent upon us, and again, this is a cartoon of what we're seeing here. It's incumbent upon us to figure out biologically why this is a problem for nephrin. But we also have to wonder, is allele-specific expression, allele-specific expression, is that a more generalized part of pathology that's contributing to kidney disease or SSNS? So to take on that project, Anna Onushik is working on that in my group, and she's been doing some very interesting preliminary work that she'll share with you guys sometime in the future. I'm not going to take away her thunder, but there seems to be evidence that there's a lot of allele-specific expression that we've never been able to look at before. And in the future, understanding that genetic architecture's contribution to disease may be very important. So future directions, as we look on the genetic architecture of nephrotic syndrome in our patients, whether here it's in Boston or down there in Brazil, the B places, Brazil and Boston, What we haven't talked about today is whether deletions or duplications, structural variation has a role in nephrotic syndrome. Likely, certainly not as much as congenital kidney anomalies, but I think there's a role. And for some of our patients, we want to make that diagnosis. We talked about whether COL4A3 and PHS1, if we're going to start to see common variants in WT1 or Poticin or other genes, whether those contribute to other forms of nephrotic syndrome. We need to just get more patients, particularly from underrepresented populations. So by recruiting more patients, such as those from Brazil, or those from Brazil, particularly from non-European populations, we're going to find new single gene causes of diseases, more loci from our GWAS studies. We're going to do rare variant discoveries, and we might find modifiers. And we're going to continue, my group is continuing to try to lead the way for facilitating data sharing through meta-analyses, additional families. We're going to continue to try to advocate for that. We're building our Big Kids biobank at Boston Children's Hospital. Right now, we're able to recruit patients in this manner across the United States. Hopefully, one day we'll be able to open this study internationally so that folks like you all who have patience and interest and excitement in doing research, and if there's capabilities that we have that you guys aren't working on, we can collaborate in that way. My belief is that as much as possible, the research should be being done in the places where the patients are, where you all are. But if there's any additional work that we can do to help in that process, we'd be very interested in doing that with you all. So I think the final comments are that, us as clinicians, there's a massive role for us clinicians in genomic discovery, because we have to have precision phenotyping just as much as precision genotyping. We need to, once we find genetic variants in known genes, we have to make sure that we're classifying genes and variants as well as possible. So I co-lead this kidney disease working group of the ClinGen Consortium, and this is comprised of nephrologists and geneticists from around the world. And we all work together collaboratively to try to understand whether genetic variants that we find are pathogenic or not, or genes. So if you're interested in being part of this, we're finding trainees around the world are particularly great in being part of this work. Please let me know. I'd be happy to connect you with folks. We have a lot of folks from the US and Australia and Europe, not that many from Asia, and very, very few from South America. It'd be great to have more representation. So I really invite you to be part of that. It's co-led by myself and Andrew Mallett, an adult nephrologist in Australia. And I think as clinicians moving forward, we need to make sure that, when we say a genetic variant is disease causing, that we're totally accurate about it. We can do more and more screening these days, and it's important that we use very, very strict pathogenicity filters to do that. I think that the work that you guys are producing and publishing is consistently highly accurate, and it's really, when you're calling these prevalence estimates and these variants as pathogenic, it's very accurate. But we need to recognize when we're reviewing the literature as clinicians and readers that we make sure that we feel comfortable with that attribution. We also need to continue to set an example for the rest of the world and our colleagues on how to do this right. And I think that our groups are two groups that are doing it right, and I look forward to continuing to disseminate that with you all. So with that, I'd like to thank you all. This is my group, and I'll stop sharing. Thanks for dealing with my technical difficulties, and I'd be happy to have a discussion and answer any questions as it gets later in the evening for you all. Thanks.
Thank you, Matthew. It's a very nice talk.
Thank you.
As always. But I have a question from the audience that they sent to me before in advance. And the question is, given the new discoveries of these common variants in steroid-sensitive nephrotic syndrome, do you think that it makes us more distant or closer to new treatments for this condition? Because when you have a rare variant of a specific disease, we have the hope of discovering a specific treatment for that condition.
Right.
But here in this situation of steroid sensitive, how far are we of foreseeing new specific treatments for this condition?
Yeah, I think that's a really important question, and I'm very optimistic and positive and happy that we're finding these genetic regions, because what we can do is we can look to other fields like inflammatory bowel disease, ulcerative colitis, Crohn's, coronary artery disease, diabetes, where large GWASs are highlighting hundreds of novel loci. And each of these loci, when biologists and experimentalists dig into the biology, they're revealing new pathways. And those pathways all present us the opportunity to try to develop rational drugs that target them. Now, all of them are not going to end up being able to be targeted for a therapeutic that could be used as humans, whether it's because of side effects or that there are very few people who have that particular genetic signature. But I firmly believe that this gives us an idea to point to rather than general immunosuppression. And finally, we can take the lesson of like a familial hypercholesterolemia, which identified the HGM-CoA mutation. But we found that statins are useful for many, many people with high cholesterol. My hope and belief is that ultimately this genetically inspired discovery will result in a class of therapeutics that are generalizable beyond those who have the signature.
Sorry, there's an helicopter here. It's Onushiki now. It's hanging.
Very nice talk, Matt. I think that you well presented perspectives that we'll have ahead of us for the following years. But one thing that I'd like to hear from you is the following. When one considers this genetic basis of nephrotic syndrome, and with the scenario that you showed, we assume that there is a significant proportion of cases that are based on a polygenic mechanism, and including small effect variants, both common variants and rare variants. And interestingly, I mean, these variants, they may well be related to genes with significant different roles, like structural genes, signaling pathway genes, genes involved in immunologic responses, and so.
Right.
So the nature of this composition of this polygenic scenario, and therefore the phenotype, may be very heterogeneous, right?
Yeah.
And, so my point is that, and I'd like to hear your perspective on, is that how much you think that that this scenario may contribute to a significant heterogeneity to immunosuppression, to response to immunosuppression. And the more you characterize this, or you have elements to do that, how do you foresee that these information can be used to predict response to immunosuppression, or at least to predict some response for attenuating, or basically showing that or predict a non-response. So
yeah, I understand.
So,
yeah, it's a really important point you're making. And I think first we should distinguish polygenic. I definitely believe that polygenic contribution is very substantial in nephrotic syndrome. But then what we can do is characterize nephrotic syndrome. I would call it a rare complex disease rather than a common complex disease, or a common complex trait. Common complex traits are things like height or obesity or EGFR or height, or coronary artery disease or lipid levels. These are traits that are incredibly polygenic, where there are hundreds to thousands of loci that all have a very, very small impact. And they combine in different ways to contribute to the heritability. Nephrotic syndrome, on the other hand, is rare complex, so it's polygenic. But what I think we're gonna find is that there are tens of loci that are associated, maybe low hundreds, rather than thousands. And what that means is that that's a track
Which would make easier to
sort out.
to look at this point that I just raised it. Yeah.
And what I think is that still each genetic contributor we can think about in terms of like, you know, our hypertensive patients, it's just a parameter. Like with our hypertensive patients, we think about their weight, their cholesterol, their smoking history, their family history. But the more that we can define those parameters, the more we can model them statistically or experimentally. So what I believe is that the GWAS will identify more and more loci, which will then allow us to categorize each patient we see, and then will allow us to statistically test whether predictive power of developing the disease in someone who doesn't have the disease, that's like a polygenic risk score, or predicting the genetic signature that's associated with immunotherapy responsiveness, or whether we develop a new drug that is directed against one or two genetic signatures, and then we see whether it's directed against, it helps in another polygenic signature. So I think there's heterogeneity inherent in this, but if we can, it's like a known it's not a confounder, it's a known component rather than unknown by more discovery. And if we know it, we can model it either at the bench or statistically or epidemiologically.
Thanks.
Yeah.
João Pesqueiro, please.
Hi, Matt.
Hi,
Hi, Matt.
good to see you again.
Good to see you again, too. Thank you for this wonderful broad view of nephrotic syndrome genomics. I have one very specific question. I was amazed by the data you showed about the STC1 gene expression between 2 risk alleles, 0 risk alleles.
Yeah.
My question, I have two questions regarding this topic. Did you check in the 1 risk allele patients whether does the expression goes is related?
Right.
And one other point, we know that the APOL1 is the expression of APOL1 is in the kidney, but it's also you have circulating APOL1. Does it apply to the expression of LCC1 in other cells in addition to
Yeah, thank you. That's really important. I have not done we did not do the 0 versus 1 versus 2 risk allele STC1 simply because we didn't have enough patients to have that be statistically powered. However, we can certainly look at that and look at just the point estimate to see if there's any intermediate effect. I'd be happy to do that. I forget, we built the APOL1 portal, and maybe, I don't know if it's possible to look at that in an additive model than recessive, but I can look at it and get back to you about that. The second question is, you're totally correct. APOL1 high-risk variant, APOL1 is expressed in the podocyte, but also the endothelium in the lung. It's highly expressed in the placenta. And as you know, there's a liver synthesized form that circulates, and that has the role in the sleeping sickness. Lots and lots of work has shown that APOL1 that's circulating is not contributing to the kidney disease, but whether there is some circulating APOL1 that's contributing to increased STC1 in the glomeruli, I don't know the answer to that. So I think that we can certainly try to test that over time, but you're right, taking into consideration like other APOL1 that's not resident podocyte to think about STC1, I think would be really important.
And another one point, when you overexpress a human STC1 in mice, you see a lot of effects, like increasing serum phosphate levels. That would be interesting to check in the patients, whether the phosphate levels, the circulating phosphate, whether it's somehow influenced by the APOL1 expression.
Yeah, that's absolutely. Like, I know of the STC1 work. There's a investigator in Texas who studied it in renal tubules and shows that it's upregulated in response to kidney injury and seems to have an antioxidant effect that's protective. But it sounds like you know of some work with phosphate metabolism and other aspects too, huh?
Mm-hmm.
Yeah, great.
Thank you very much.
Yeah, you're welcome. Absolutely.
But we are all eagerly awaiting for the results of Apollo study.
Yeah.
And it's ongoing for such a long time. And I would ask you today, if I have a donor who is who has one risk allele of this APOL1 gene,
Yeah.
and there are two recipients, a child and an old guy. Would you use these criteria to direct the kidney from this donor to the old guy instead of the child, or would you ignore it completely?
Yeah. Well, one thing is I would never ignore it. I would just consciously choose to not characterize it, And secondly, I'm not a transplant nephrologist. I'm not involved in those decisions clinically, but the question really is that considered more of a risky kidney for long-term outcomes? And what I would say is thus far, we have not seen, that I know of in the literature, we have not seen any evidence in delayed graft function or, ultimately, loss of graft associated with one risk allele. So I would not, from my knowledge, I would not direct it towards an older person based on just one risk allele. Would anyone do anything differently on this call? Any of the transplant nephrologists here?
No.
No. Okay.
But you know that everybody is awaiting for the results of this study.
Yeah. Yeah, I wonder over time, I really wonder if you take half the nephron mass away from someone who has two risk alleles, is that sufficient to kick off an APOL1 associated kidney disease? And if so, what are the odds? And then does a donor have a choice of whether or not to have increased odds? And and then there's some indication that donors recipients with two risk alleles are not thought to have any increased risk of graft function, but there was a recent paper, I think in JCI, that suggested there was perhaps a little bit of increased risk. So this is where being a really good epidemiologist and reviewing the literature to understand how this study is being designed is really important, and interpreted is really important.
Matt, may I ask one more question?
Yes.
Please.
Regarding the allele specific expression.
Yeah.
Did you think of stability of the risk allele? Whether the risk alleles are less stable?
Yeah, exactly.
Less stable.
Yeah, that's a hypothesis that we need to test. Is somehow there are these synonymous variants that we think might be the functional variant that's leading to the allele specific expression, and is that somehow altering mRNA dynamics and ultimately in a pre-protein level for sure. So there's a lot of essentially a brand new concept. So we have a lot of work to do, and by we I mean Anna.
That's very interesting.
Do you think that animal studies could help on that?
Which study?
On allele stability analysis.
Yeah, I think it will help. I think it will be very helpful to do that for sure.
Mara.
Hi, Matt.
Hey, Lara, how are you?
I'm fine.
Great.
I would like to say for me and for Vera Belangeiro that it's here. We are very pleased to hear about nephrine studies because I got my PhD in nephrine and potassium mutations.
Oh, great.
A long time ago, yes. And Vera always told me that the steroid sensitive individuals should have polymorphisms or risk alleles, yes.
Yeah.
And I believe she's very happy today. Yes. Because we believe we have 100 patients that she is the doctor. I am a biologist, yes.
Right.
And we have many polymorphisms, yes, SNPs.
Yeah.
That we believe that are doing something in the context of sensitive, yes.
Oh wow.
Yes.
Well,
but we have to prove it, yes.
Yeah. Well, I would love to have a further conversation with you all about this because on a couple levels. One is we're in search of mechanism, and it'd be great to understand the mechanism, and great to have as many people work on this as possible. The second is that this risk allele, these risk variants that we found, are very specific to East Asians. They're Japanese, Korean. There's actually a hint in Africans, sub-Saharan Africans as well. And we think that if we had more African patients in the study, we might find a variant there. But it'd be for instance, we really don't see a signal in European patients, but it'd be very, very important for us to look and think about what is the spectrum of allelic variation in nephrin in these regions in your patients, because it may be that we'll certainly get more insights on whether there's a shared haplotype with the East Asians or whether there's this African-specific variant. And then to be able to, with the Neptune data we have glomerular transcriptome data, epigenetic data that's coming out of our group. I think we should be able to help you guys try to understand the potential functionality of the nephrin. But I just had a postdoc arrive from Zurich, who's a biologist, and I'm putting her on this project to try to elucidate what the biology is here. But the more we can talk about it and work on it together, I'd really love that.
Thanks for the talk.
Yeah, absolutely. You're welcome.
Okay, I think we're done now, Matt.
Okay.
I would like, again, to thank you, to say that it's always a pleasure to discuss with you.
Yeah.
I think it's a very hot topic. There are many questions and this is good because makes us alive.
Very much so.
Searching and researching, okay?
Yeah. Alive, and like the slit diaphragms, we're interdigitating again.
Yeah.
Being my REBRASNI friend. So it's good to be back and connecting again. I look forward to seeing you guys in the future, and I'd be happy to come back and chat another time.
Okay, thank you.
All right, good night.
Thank you, Matt.
Thank you.
Thank you, Matt.
Good night.
Thank you.
Good night.
Vou interromper a gravação aqui, porque senão fica gravando, a gente sai daqui da sala e fica gravando. Interromper a gravação. Stop. Ah, foi legal, né?